So when we were planning for this panel, we realized we could spend the entire day talking about early endpoints, but we only have an hour, especially if we want to get to your questions, and I will try and get 10 minutes at least for questions. And so we're going to focus on endpoints applied early in the course of disease when it is still curable. It is easy to be in favor of accelerating the pace of learning and to more rapidly identify more effective and less toxic therapies and getting them to patients sooner and being able to adapt therapy on that basis to work on curative treatment rather than just extending by several months of time. However, how and whether this can be achieved is what is at issue. And so we have really a great panel today that has brought us a lot of expertise from all sectors in the conversation, as Rick Pazdur has always said we should do. So we have patient advocates, regulatory leaders, the leaders of our government investment portfolio in cancer, pharma, payers, who actually ultimately make the decision about whether drugs should go on the formulary, academicians, and clinicians. So we hope that this conversation will allow us some insight and a commitment for how to work harder on these issues that truly can transform the pace of drug development. So I want to start with the panel. We have, from the regulatory perspective, Dr. Richard Pazdur, who came to the FDA from MD Anderson in 1999 to lead the Division of Oncology under Cedar, and then was the inaugural director and founder of the Center of Excellence, Oncology Center of Excellence at FDA, and for the last 2 months has been leading Cedar until his retirement just a few days ago, which we hope will be temporary. Okay, so from the academic perspective, as well as from the person... the person most able to set priorities in the investment in cancer, we have Dr. Anthony Letai, who joined the NCI as the 18th NCI director in September of 2029. He was previously at Harvard Medical School in Hem-Onc at Dana-Farber, researching treating hematologic malignancies, advancing biomarkers to guide treatments and therapies such as BCL2 inhibitors, where they have become the standard for leukemia treatments. We're very fortunate to have, from the health plan perspective and the payer side, Dr. Kate Goodrich, who is the executive in residence at Optum Healthcare. She's held many leadership positions from a payer perspective in health administration, including the CMO of Humana, as well as from our largest payer, the Center for Medicare and Medicaid Services, where she served in the role of CMO, as well as the Director for Clinical Standards and Quality, and oversaw many value-based programs. From the biopharmaceutical perspective, we have Hal Barron, who's been a leader in the pharmaceutical industry and served as the CMO for Genentech, where I first met him and we began working on early endpoints for accelerated approval in the high-risk early breast cancer setting. He served as president of research and development at GSK from 2018 to 2022, when he then switched to become the CEO of Altos Labs, which he'll tell you a little bit more about then. But more importantly, he is also a card-carrying cardiologist, perhaps the field that is best to use early endpoints to transform and accelerate learning in that field. I already introduced Keli Santos-Parker, as you heard, and I'm Laura Esserman. I lead the breast cancer program at UCSF and have led the I-SPY 2 platform trials for neoadjuvant therapy for women with high-risk breast cancer, so we can accelerate the pace of finding the right drugs for the right person, and we've extended this model to other cancers as well as COVID and ARDS. And I helped found a nonprofit, Quantum Leap Healthcare, to enable patient-centered dynamic trials to be to accelerate the pace of learning in trials and make them more efficient, even before AI. I want to introduce my co-moderator, Jane Perlmutter, who is a patient advocate exemplar who, in her former life, was a psychologist and the president and founder of the Gemini group. And fortunately for the community, she has had a tremendous influence on so many organizations, from cooperative groups, the DoD, PCORI, and I-SPY, and has channeled her energy into making a difference for patients and insisting on patient-centered trial designs. Jane.
So first of all, thanks so much for including me, and especially as a co-moderator. I think I'm the only person sitting up here who is specifically thinking about patients, but I think most of you either have been patients or remember patients, and particularly for this talk about or this panel on early endpoints, please keep them in mind as well. I know all of you, and what we're all here about is improving patient outcomes. But it's complicated. I like to say not all patients have the luxury of patience. So, just thinking about safety is not always necessarily the right way to go, because we would never make any progress if we were that conservative. And the other thing I like to say is all patients have the same goals. They want a long and high-quality life. The rub is that what many patients think about that. They have a variety of opinions about the trade-offs and what it means to have a high quality of life. So I hope here we think about not only safety in terms of serious adverse events, but tolerability. Many patients I know have talked about they'd rather be in the hospital for a couple of days dealing with a serious adverse event than having a long-term low-grade toxicity, fatigue, or diarrhea. I think we need to keep that in mind as we're thinking about what's in the best interest in patients, and I hope that we will, and we keep that in mind as we think about early endpoints that will get treatments to patients when they need them, even if there is perhaps a little risk.
Okay, so we're going to start with the example of cancer, and I want to start with the so-called neoadjuvant therapy setting, because that's where systemic used to be. As a surgeon, I can say this, that we used to start with surgery first. That really, in my opinion, is the wrong way to do things, because surgery doesn't save people's lives. As if people are at risk for metastatic disease and they present with high-risk disease, systemic therapy is what will change their lives and allow them to survive. But it also gives us an amazing opportunity to evaluate these early response markers. And it also means that people will have less morbid surgical procedures, which can make a huge difference. So it allows us both to de-escalate and escalate appropriately if we have measures that we can truly believe in. So starting with biomarkers, I think there is, I think these can be a little bit confusing. So there are two types of biomarkers. There's the response biomarkers. That's whether the tumor goes away, as Keli was talking about, complete response or near complete response. Whether that's measured by pathology, whether it's measured even earlier than pathology, looking at ctDNA or functional tumor volume. But then there's also the biomarkers that help you more properly bin patients and figure out what the target should be, and how to divide up the population, which not all pharma companies like, but they have a value for defining these early endpoints. They're related but independent. So I want to start with, Rick, maybe really talking about what do you think the role the FDA can play in encouraging the collection of data that really helps us to validate these early endpoints and to make this really a part of what we do?
Well, I think, when we take a look at multiple endpoints that we've developed over the time, they were a collaboration with the community, so to speak. And that's one point I really want to get across, that it's not just the FDA saying, this is going to be the endpoint here for drug approval, that we really take a look at really bringing in the community, building consensus. And you could see how we did this with multiple endpoints, most recently with the minimal residual disease endpoint in multiple myeloma. This was a long process. But here again, Laura, I think what is important, and you are pointing it out early in your previous comments here, what is the purpose of the endpoint? If you're looking at an endpoint for drug approval, obviously that's going to have a much more robust data package that's associated with it, the buy-in of the community that this truly has a relationship to the ultimate clinical endpoint. If it's more of an exploratory endpoint, such as a possible predictive endpoint, the onus is really almost on the pharmaceutical company. Do they feel that they're comfortable in leveraging their development plan on the basis of that endpoint? So it really depends on what it is. One point I really want to get across, because I don't think, and this is applicable to this disease setting, but also even in the disease setting of metastatic disease, is that we cannot divorce the discussion of the endpoint and efficacy endpoint from the safety endpoint. And we've seen that in looking at response rates in metastatic disease, where many companies push the dose of a drug to get the highest response rate, and they're not aware that subsequently they have tremendous dose modifications. Then when you take these drugs out of the early drug development and put them into a randomized setting, because there are so many dose modifications afterwards, they actually lose. So really, we can't divorce the issue of efficacy from safety here, and I think that that's an important point.
I think it's a critical point, and I also think that goes to how long people should remain on these drugs. And again, these early endpoints tell you when people are doing extremely well. So now you're taking a toxic drug and continuing it. We need to really find a way not to incentivize the continuation of drugs that can.
Laura, as you know, from a pharmaceutical point of view, most of our trials now are looking at longer durations of treatment. They have not answered questions of phase of contribution, so to speak. So there is a problem here that needs to be addressed.
For sure. Okay.
And I just have to mention the fact that we never look at late occurring side effects that extending the drug too long is likely to.
Yes, absolutely. So, Tony, I'd like to turn to you. So what about the NCI? What do you think the NCI can do to advance biomarker discovery and validation?
I think, well, as you probably know, most of the support that NCI gives to the sort of National Cancer Project is via our extramural research programs. And of course, we support a wide variety of clinical trials. We also provide in-kind support in the intramural program for some of these clinical trials, including biomarker development, biomarker validation. But we also provide resources in money terms. One of our flagship efforts is the assay validation of high quality markers for clinical studies in cancer, which is a funding opportunity that was just recently reissued, but it's been available for several years. And that studies both toxicities, that helps the development, both the preclinical analytical development as well as the clinical development of biomarkers, largely directed at looking at effect in terms of pharmacodynamics as well as toxicity. So the NCI is very well aware of the benefit that really good biomarkers can bring to improving clinical trials and clinical care, and we have several ways of doing it.
Great. So, Keli, is there a way to integrate both types of these biomarkers, or to have the math help you understand how one can inform the other?
Yes, and I think that the classification of biomarkers can be quite complicated, and the FDA has a lot of guidance on that. But generally, thinking about time and a patient's experience and what that patient is going to experience through their course of clinical care, we really emphasize. I just want to go back to something that was just said, which is the development of biomarkers is so critical. And the thing about an early endpoint or an early biomarker is you had to measure it early on. You can't go back in time and measure that. And so one of the things that's really incumbent on us as we design new trials and as we continue our research moving forward is we're not just looking for new drugs. We're not just trying to understand the mechanisms, but we're applying those mechanisms to try to understand what we should measure to quantify this response. And we need to do that early on. And then to answer your question, Laura, then when you have that outcome much later, you can retrospectively go back, look at your patient cohort, ask, how did all of these details come into play mechanistically to explain these outcomes that I'm seeing later? But you have to have that foresight to measure it early.
Okay. So, Hal, I'm going to put you in the hot seat. So, what is your prescription for how pharma can help us get away from one drug for everyone and think about applying drugs to more subsets? We talked about this even in the AI panel. Oh, AI can find the subsets that work. But even if you find them, is that going to make, what's going to make pharma want to invest in these smaller subsets or figure out how we're going to do that? So if we have these really early endpoints and we know these, we can identify these smaller subsets, how are we going to push that forward?
Well, I'm not going to speak for all of pharma, but I can tell you that it's incredibly intuitive, and there's a ton of data to suggest that using biomarkers to stratify patients or to identify when drugs are active is extremely cost effective, is very, very good for patients. It's very good for pharma. There's so many examples I could give where the pharma has done this well and can prove that it was not only faster, cheaper, better for patients, but much more financially. When you, let's just use HER2 and breast cancer, 25% of women with breast cancer have HER2 positive. If a drug like Herceptin had been developed for 100% of patients with breast cancer, it would have failed, clearly failed. So when you do the math on, I could treat everybody and charge blank versus I can treat only 25% of the people and have 1/4 the revenue, you're missing the fact that it didn't work without the stratification. And that's been borne over and over and over. So I actually think personally that for most, and I'll just use the term enlightened pharma companies and biotechs, this is well ingrained in everything they do. I think the biomarker discussion is challenging though, because we don't have always biomarkers that are, many biomarkers are prognostic, meaning they predict outcome and severity of morbidity and mortality, but many are not in the causal pathway. And, we use examples like cholesterol, which is predictive of cardiovascular disease, and lowering it reduces cardiovascular disease. Hemoglobin A1C is predictive of cardiovascular events and lowering it. But, if you look at the number of PVCs, premature ventricular contractions a patient has after an infarct, it's extremely predictive of overall survival in the first year. But we've known from randomized trials that lowering that doesn't improve outcome. And so that's a good prognostic biomarker, but it's not predictive of a treatment effect. And so you need to spend a lot of time and money to actually collect the right biomarkers. And when you do so, not only do you deconstruct the heterogeneity of disease and identify ways of determining whether your drug's active in a much faster, shorter, cheaper time frame. If it doesn't work, and 75% of phase 2s fail, so it'd be terrific to figure that out faster because, people quote, and it was mentioned in the last thing, that 90% of drugs fail, which is true, but the vast majority of expense is in phase 3. The failure in phase 1 and 2 is acceptable if you never fail in phase 3. So the key thing is, how do you collect enough biomarkers to kill your drug at the end of phase 2 or advance it aggressively in phase 3? And
in the right population.
In the right population. So if you then understand that not only can you use it to stratify which drugs are likely to work in a Bayesian way, if I thought it was somewhat likely to work, I'm much more likely to work at the end of phase 2, as Rick's point. But then also there's the possibility that you can use the biomarker as a predictive tool, much higher bar, but does the biomarker at baseline predict, like cholesterol, for instance, predict who's going to benefit more or less? And then you can start stratifying the disease, enriching for
within
people
the subtypes,
within subsets. And even in the extreme, you can look at the different biomarkers and try to understand the pathophysiology of disease to find even more targets that maybe you weren't even pursuing. So if your biomarker is a series of inflammatory cytokines, that when they are suppressed, the disease gets better, those cytokine suppressions can end up being targets for the next drug. So there's so many different ways biomarkers, when they're augmented in clinical development, can not only increase the probability of success, identify the right population, ensure you don't go to phase 3 with drugs that won't work. And I think personally that for enlightened pharma and biotech, this is well understood and routinely done.
Richer, better, more instructive phase 2 trials in the early disease setting to prevent metastatic disease. Rick, you wanted to say something before you get to Kate.
Companies not doing this, okay? And that, I think, is an important point.
They're
going for an ITT population when they specifically know the drug works in a marker-positive population here. And I think it's really incumbent for companies to realize that patients understand this, that they are marker-positive, marker-negative. They're looking for what is my treatment effect given the information that I have at hand, so to speak. I think a classic example of what you're referring to, I went back to the archives of oncology, is the development of ERISA, where they had a general population of non-small cell lung cancer patients, 5 clinical trials, large clinical trials that were completely negative, only to have other drugs come out looking at the specific target get approved. The drug was subsequently resurrected, but had a tremendously flawed development program because they did not take advantage.
But everyone here is going to take this to heart and change their development process.
Yes.
Okay. So, Kate, I want to ask you, so what the payers and the health systems are really looking for, what to pay for biomarkers? What's the kind of proof that you're looking at, since you're advising? You're often in a position to advise that. What are you looking for?
So I think I want to start by maybe referencing my experience working at CMS, where part of my work was focused on making decisions around what Medicare will cover and pay for. And this is important because many payers, if not most payers, track what CMS does. Either they directly will have the same policies, or they will look to see what the agency has determined through national coverage determinations or other types of coverage determinations. So one of the challenges that CMS faces is that the requirement for a positive coverage determination by Medicare is very different from the requirements for approval by the FDA. They rely on different laws that were passed by Congress. So while the FDA is, of course, focused on safety and efficacy broadly and has naturally increasingly used earlier endpoints to make approval or clearance decisions, the bar is just different for CMS and therefore for most payers. CMS has what's called the reasonable and necessary standard, which has been translated into a focus on health outcomes, whether that's defined by a hard outcome that's related to mortality or morbidity, or also often quality of life. Over the years, as again, FDA has relied in certain circumstances more on earlier endpoints, that level of evidence is typically not enough for CMS to make a positive coverage decision. However, when I was at the agency, we knew that a lot of these drugs, devices, diagnostics were had a lot of potential for really benefiting patients, so the agency developed a number of sort of instruments, if you will, to conditionally cover certain technologies or even sometimes drugs when there wasn't enough evidence to demonstrate a particular health outcome, but there were positive signals through early endpoints. So you all may be familiar with coverage with evidence development or FDA CMS parallel review. These were attempts to allow the agency, and often therefore other payers, to cover a drug or a device or a diagnostic before harder outcomes were realized, because those, of course, take so much longer. And so what that requires typically is for a manufacturer to continue with a type of clinical evidence generation, whether that be ongoing randomized controlled trials or often a patient registry. And then over time, once clinical endpoints were met, later endpoints, the agency could determine whether to continue coverage or to rescind coverage depending upon those endpoints.
So that might be also that people would be, as a community, more likely to do approvals with early endpoints if we do the hard work of collecting the data very carefully, with the willingness to, as you say, Rick, rescind the drug if we don't meet those measures. Okay, I now have truly a 10-second answer from everyone, because I think it reveals the things that we're all worried about before I turn it over to Jane. So I want to know, when we think about early endpoints for regulatory approval or payment or for moving it forward to the clinic, each stakeholder is likely to have different concerns. So I want to know, what keeps you up at night about this? What is the mistake that you're worried about, Rick?
The relationship of the earlier clinical endpoint to ultimate clinical benefit. Okay. And one thing that we have to really emphasize when we talk about these earlier clinical endpoints is the magnitude of effect on that endpoint. Okay. Yeah. And I think that has frequently been ignored here. When one has a tremendous big effect on an endpoint, there's much greater confidence from a regulatory point of view of extending these forward.
Okay, Tony?
Pretty much agree with Rick. There's a lot of early endpoints that make a lot of sense that won't necessarily give you the OS or DFS endpoint that you're really looking for.
Okay, Hal?
Yeah, I agree. And we'll say it slightly differently, is the biomarker in the causal pathway.
Keli?
I think one of the questions we had earlier today was talking about, are we measuring these things in the correct patient populations? And that's what I worry about, that we're not measuring the correct things in the broad population, all the way out there, everyone who could potentially benefit from these things we're developing.
Kate?
Yeah, payers have the exact same concern, the relationship between early endpoints and later endpoints. So
Yeah.
it's already been said.
I would say, for me, the fear of, fear that we might make a mistake that doesn't allow us to really move forward quickly. Especially if you're willing to collect, if you have good enough data and you're really willing to set up a system to carefully collect and validate. Jane?
So what I'm hearing a lot of is a real risk aversion, and I understand we don't ever want to be wrong and approve something that doesn't turn out to be right. But particularly, Rick, when you talk about you want big effects, at a time when there were very few drugs, we were looking for small effects, whatever. But increasingly, we have more and more drugs, particularly in cancer, but I believe in other areas as well. And even the best drugs don't work for every patient. And even drugs that have a small or a big efficacy effect have some toxicities. And I think from patients' point of view, they really want to have more options. They want to have an option if this drug fails them to have a different drug. If this drug has a toxicity, one that might be a little bit less efficacious on the mean could still help them. My sense is that FDA has been pretty paternalistic in their
I used that word.
Right, and
I think I used that word in our call.
and I said, well, I need to quote that
Set herself up for it.
because that's exactly
That was my feeling.
Right.
Here again, I want to tell people how this kind of concept evolved between effect and toxicity goes. And subsequent drug approval. We didn't want to get into a situation where we were approving drugs that had less efficacy because you could, over time, just start approving placebos, basically. And that's why initially, about 20 years ago, we had discussions about using time to treatment failure versus progression-free survival. And we went with progression-free survival because it was an efficacy endpoint. Time to treatment failure had a toxicity element. So if you develop drugs and are approving drugs with less and less efficacy, you could go down that pathway. But that's an extreme. And can you have a situation where you have a less efficacious drug? But that requires, I think, discussion with the community again, that this is an appropriate thing to do, in what context it's being used, especially in late stage disease. But that has been the concern. Are we going to go down this garden pathway of eventually approving placebos? That doesn't necessarily have to be, so to speak.
I mean, there's always a slippery slope. But
and here again, from a regulatory point of view, remember, the drug has to demonstrate safety and efficacy. It doesn't have to demonstrate basically better efficacy and better safety, so to speak.
But it seems like that's the way.
And that's why we wanted to have non-inferiority trials and went down that garden pathway for many years and still do, obviously. But these are very complicated trials to do with large patient populations. So most people want to do a superiority trial.
Right.
Nobody wants to go a priori to demonstrate that their drug is inferior to a competitor.
But they'll be superior if they're
better safety.
Less...
But that's a nuanced discussion that we haven't had. And many companies that have failed trials make that argument. And when you actually take a look at the safety, they're not better safety, they're a trade-off of safety, so to speak. And that's maybe important to certain individuals. But truly, if you had a markedly different safety profile, that I think would deserve
and that would constitute a similar efficacy and much better tolerability is superior, right?
From a
potentially.
benefit
But
analysis.
then we have to force ourselves to really refine the measures.
But traditionally, we've looked at the demonstration of efficacy and safety in two separate analyses.
We keep coming back to the safety, and I want to keep coming back to tolerability. And one of the problems is we don't often collect adequate information about how patients feel and function. Actually, let me ask Tony. I've been involved in what used to be the cancer cooperative groups for more than 20 years, and it's been a real push of advocates to ask for patient-reported outcomes. And even now, it's very rare that the NCTN trials in cancer include those. Why is that the case? What is what is NCI doing to make sure that we know about tolerability of drugs?
So, with regard to patient-related outcomes, it definitely is an interest of the NCI. The NCI has done several things with regarding that. It developed the Pro CTCAE, which is sort of a
About 10 years ago.
standardization of patient-related outcomes. We have a tolerability consortium that sponsored a special issue in cancer just in 2025 that was all about patient-related outcomes. A lot of our trials do record patient-related outcomes. I can't say on a case-by-case basis which do and which don't. I do want to make the point that when we don't that the difference between patient-related outcomes, if a study isn't reporting those, it is reporting adverse events, and there is some overlap in what is being measured. A lot of what's different is how it's communicated. Patient PROs being related more directly from the patient, and AEs coming through the clinician. But similar things like nausea, diarrhea, fatigue get recorded in both regimens. How it's reported is what is significantly different. We rely on the investigators when they're designing their trials to prioritize the information that's going to help them with the decision that's going to be most pertinent with respect to their primary outcome measure. So we so far have not chosen to impose, sort of with a heavy hand exactly what is needed for that. And we have to trust our clinicians to measure what resources are most useful for their trial.
I think it's interesting that the patient-reported and physician-reported and outcomes are really not necessarily the same. They come out quite differently. So I think it's incumbent upon us to really push ourselves to make sure we do measure both and then start incorporating this into trials and do a better job of being really more rigorous about fewer and more important things.
Right. And we're often told that NCI doesn't have the budget for patient-reported outcomes, but I urge all of you industry folks to include them. And I want to ask Kate, how do the payers react to data when they have data about tolerability? Is that something that would be important for them to consider when they're agreeing to pay for a drug for a patient?
No, this is a really important question. I first want to start by maybe dispelling a myth that I often hear about payers and how decisions are made around coverage at CMS, but also at every payer that I'm aware of, including where I work now and where I've worked previously. Decisions about coverage of a drug or a other type of clinical service are made by clinicians. So for drugs, this is a combination of pharmacists, physicians, and nurses. These decisions are not made by the CFO. So as such, payers are, in fact, quite focused on toxicity and very attuned to toxicity. It's always a very integral part of the clinical policy conversation because obviously it's terrible for patients when there are severe adverse events or the ones like you were mentioning, Jane, that's sort of the lower grade long-term toxicities. And because, of course, toxicities can lead to higher costs for payers and for patients, such as ER visits, hospitalizations, other drugs to manage the toxicities, et cetera. So the evidence reviews that our clinicians undertake always incorporate toxicity into the decision making. So that will affect not just whether or not a drug should be covered, but also where it might belong on the formulary. So, for example, an equally efficacious drug with lower toxicity may belong on a lower tier than a more toxic drug that the evidence shows leads to more complications and serious adverse events. And I do also want to say payers are also extremely interested in patient-reported outcomes. I just want to say that because that was just mentioned. And particularly when there can be some consistency for a particular disease or syndrome in the PROs that are collected. So it makes it easier to compare from one study to the next. But that is also really, really important to us.
Thank you. I'd like to turn a little bit to innovative trial design because I think ultimately that's where these early endpoints are going to play a role, and that's where we're going to lead to better, faster patients treatments. But Hal, during our planning trial, you talked a lot about a response discontinuation trial design. That's one that I saw many, many years ago and liked, but haven't seen much of. Could you make sure that we all understand what that is and what the advantages of it are?
Sure. I mean, of course, biomarkers have so much value in so many different things. One of the more aspirational aspects, I think, of biomarkers is that we always strive for a baseline covariate that is a predictive biomarker. Like I used HER2, you can use lots of things that identify patients who will and therefore won't benefit from a drug. And that's terrific when you got, EGFR overexpression or mutation, HER2, all the different baseline covariates that are what I call predictive biomarkers are altering the treatment effect. But because we don't have the data and profoundly don't have an understanding of biology, oftentimes the baseline covariate that predicts the treatment effect is missing. And it may not even evolve until your post-phase 3 failure. It may never evolve. But one of the interesting lessons I learned in cardiology was that certain fields that don't have a biomarker for treatment effect modifier or predictive biomarker use the drug as a treatment assessor, as a diagnostic. So in other words, if you come in with hypertension, people don't do a renin aldosterone workup and see if your endothelium is more sensitive to calcium or whether you're more responsive to a diuretic. You get a drug. If it works, after 6 weeks, you keep it. If it doesn't, you go to the next one. Now, you might say that sounds a little naive, but I'll use the drug that works. And that can be translated into a clinical trial design where if you have a biomarker that is in the causal pathway, you can imagine randomizing people to treat everyone with a drug, see whose biomarker changes, exclude those whose biomarker doesn't reduce because the disease is very heterogeneous and you wouldn't expect everyone to benefit. So you take the 20, 30% of patients whose biomarker improves dramatically, and then you randomize them to continue or not to continue. It doesn't lend itself well to certain modalities like AAV or maybe even antibodies that are long-acting, but for small molecules, it's very easy to have a washout period. And what that does is use the drug's effect on the biomarker as a way of stratifying patients because we don't understand the biology behind the disease as well as we'd like. So that's been termed a randomized discontinuation. It's rarely used. It's used in inflammatory bowel disease but it's not called that. It's the induction versus the maintenance phase. Hypertension doctors wouldn't say they're doing randomized discontinuation, but essentially that's effectively what's going on. And it's done in clinical medicine all the time. In psychiatry, if a drug for depression doesn't work, you can try the next one. We don't try to figure out, are you with serotonin, or are you norepinephrine or dopamine? The biology is just too complicated. We're too far behind. So someday I hope that we can use biomarkers not only
We can
to...
do it now.
We can do it now, yeah.
It's really, you set me up perfectly because so many patients, certainly cancer patients, but anyone with a chronic disease, is you try something, you eventually try something else probably. There is a design for that. It's called a SMART trial.
No, well, but the key thing here is, and it gets back to what Rick said in the beginning that everyone agreed with, if your biomarker is not in the causal pathway, you may be misleading yourself as to who's benefiting. You really do need a biomarker that you're confident when it's not lowering, that drug's not working. And that's what we're missing for many diseases. We don't have the same degree of confidence.
But even if you don't have a biomarker to predict that, you can look at the response as a biomarker. So Keli, tell us about what sequential multi-assignment randomized trials, SMART trials, where we have to go in the future, although I know Rick hates anything that's complicated.
Simplest is always the best, okay?
Not necessarily.
Well, I think Hal just said that that's a pretty simplistic way to do it, right? Try one thing and then try something else. And what you're referring to with a SMART trial design is a design where you're actually setting up your statistics from the beginning to hopefully have a method to compare these various arms and these various treatment strategies at the end of the day. But also, Hal, you said something at the end of your point there, which was, is your biomarker in the causal pathway? And I think that's why we keep coming back to patients and we keep coming back to patient level data and capturing more data, because if we don't understand the mechanisms, we won't understand why we're seeing the outcomes that happen. But those all come from patients. And so, I showed that video on my little talk with that arrow way at the beginning. As that Kaplan-Meier curve changes, which a lot of us are used to looking at those curves in order to make decisions about how things worked, every single one of those decreases in that line is a patient who died on trial. Now, what about everybody else in the population who didn't even have a trial? So this is why it's so critical that we are collecting patient data, collecting it sequentially, and then actually learning from these sequences of treatments that are going through, not just in the trial standpoint, not just in the SMART standpoint, but also the real world where patients are experiencing the diseases.
Yeah, let me just clarify one point just so it didn't get lost. It's possible that if you give an antihypertensive and the patient's blood pressure doesn't go down, that it would still reduce cardiovascular risk, but it's very unlikely. We understand the biology. So you move on.
You can go
But in-
with a probability of that. But that's just like if the disease disappears in a cancer setting, probability is extremely high that it's going to work. And you can sort and the subtypes help you sort out those biomarkers.
disappears. But my only point I wanted to make was if you don't, quote, respond, but you have a 29% reduction versus someone who responded who had a 31%, I'm not convinced that's terribly different. So I think you have to make sure that your algorithm and your endpoint, your biomarker, whatever it is really robust to be able to make that kind of comment.
Correct. And that's why we need to collect the
That's what
data.
we're missing.
And that's the task that Rick has given us. But we look forward to showing you that
By all
we'll have
means,
the
yes.
data. Right?
Also, the thing we have to point out, it is many of the endpoints that we have in oncology are probably not in the causal pathway, response rate, progression-free survival. And we saw that, we got lulled into that comfort with cytotoxic therapies. But when one took a look at the immunological therapies, especially the PD-1 drugs, you saw survival advantages with the checkpoint inhibitors that were really disassociated with response rates and progression-free
But also associated. So they really accelerate the chance that they double the numbers, triple the numbers,
we
right?
drugs on the basis of those endpoints.
Right.
They did have some response. And here again, some of the endpoints that we use when we talk about earlier clinical endpoints, are they really endpoints that we should approve drugs on? We went down that garden pathway with response rates giving full approvals where we couldn't do randomized studies because we believed that was the practice of oncology, that in general patients benefit when they're responding, okay? Although it's hard to do, to actually verify that. But in clinical practice, that's what would be done.
Right. And so we need trials that look more like care and care that looks more like trials, and that is what AI hopefully will help us move towards. So I think
that brings us to large, simple trials, by the way.
Large, simple trials, but simple can also be is someone responding to something, so you give someone a chance to try something less toxic or simpler, or, and if you don't respond, no harm done, move on to the next thing. I think that's really what the opportunity of early moving drug development to the early setting.
It's not simple. Really.
If I
Care
could
is not
just
simple.
It's not. But when I talk about large, simple trials, I'm talking about reducing the amount of data that is collected. I'm talking about looking at hard clinical endpoints, trials that resemble the clinical practice of medicine rather than the artificial trials that we do that have very specific patient criteria that do not represent what the ultimate patient population
I think the coming AI that we should be aspiring to collect the kind of data that helps us. It's going to be much easier to do, and that should be part of practice.
And my goal and my dream would be to have
RWE, right?
Huh?
And you're a big fan of real world evidence that is more sophisticated.
I would like to have these trials done in electronic healthcare systems and the records there, and have the randomization done there, and have all the orders written in these systems, as well as the data collected and then exported into a.
Is that what you're going to do now that you're retired?
I am not retired, by the way.
No.
I am not going to a nursing home, Jane. Okay.
We certainly hope not.
I mean, you're next.
All right. Okay. All right. Before we go, we're going to open up to some questions in just a second. But I just want to very briefly talk about prevention, or, prevention of aging or prevention of cancer. You cannot run these kinds of trials without early endpoints. We just successfully ran a, personalized screening trial, and we did it in 7 years at like a fifth the cost, because that's how PCORI was set up. It's possible to do these things, but again, we have to really think about the endpoints. So you change fields, right, to invest in the science of aging and buffering. So how do you see the science converging here?
I changed fields because I got older. Yeah.
That's the good news.
That's
with
the good
cancer
news,
patients.
exactly.
Well, I think young people with cancer want to get older. Yes.
Yes.
Cool.
If you look at disease, almost every disease, either the first or the second most prominent risk factor is age. And because time isn't modifiable, it hasn't been like cholesterol, we've tried to reduce it. But we've taken a different view that says that maybe some of the pathology that occurs with time might be a confounder, and that it's the pathology that develops over time that can be modified. If one is to imagine that's possible, then if you were to develop a drug that would modify that time-dependent, what I call buffer capacity loss of a cell or an organ, you would need a biomarker to figure that out, because a trial that might look to see if you extended life by 3 or 4 years, which would be more profound for our healthcare system than curing quite a few diseases, would probably read out after the patent expiry. And it would also take 30 years and not benefit most of us in this room. So the point is that as important it is to find the right targets for this buffer improving concept, probably equally important is to find a biomarker that reflects your a biomarker that's in the causal pathway that reflects whether you're in fact suppressing this pathology, and in whom are you suppressing it, because the chance that we're all aging from the same problem is almost zero. I mean, some of us will have profound muscle weakness and be sharp as a tack, and other people will be doing gymnastics and can't remember where their keys are. Everybody ages in a different way. And so this whole concept that I just described for the value and opportunity that biomarkers provide, but the challenge, of course, and it was said in the prior, this is all about massive amount of data to be able to understand the pathophysiology of aging, the biomarkers behind that. This is no small feat. And so
well, maybe how we should be thinking about looking at somehow some of our very toxic side of chemotherapies, accelerate aging. And if you can show that you're not doing that with some of the new drugs, that could be a superior. These are opportunities, yeah.
As instead of using prevention, let's use risk reduction, okay? Because I think that's what we kind of are talking here, and came out when we did the tamoxifen prevention trials, et cetera. But I think the drugs have to be very well tolerated. Okay, if people are going to be on them for long periods of time, they have to be generally, people have very little side effects or virtually none. There may be some people that do have side effects, they'll come off of it though. And you see that with statins, and you see that with antihypertensives. But also, you need a biomarker that patients can see that they're getting benefit from. When you're taking cholesterol-lowering medication, people go on that because they see it's coming down, your LDL is coming down, and therefore they want to stay on it. I think one of the problems that we had with the tamoxifen risk reduction,
100%.
it was like, I'm taking this, I'm getting some side effects, am I benefiting from it? I don't know.
Yeah. Having run a prevention program for 25 years, I can tell you that is one of the great potential areas for the imaging biomarkers. You can tell in a month.
But you need to have the patient realize that they're getting
correct,
benefit
and
from
they
it.
have to be,
and they have to be able to maintain, a normal life.
Yeah.
Okay.
Absolutely. So Tony, I just wanted to turn to you for a minute, then I'm going to open up to questions. This is a potential area that the NCI could really start investing in, really trying to figure out how do we start developing some of these biomarkers, risk reduction biomarkers. What do you think? Is this an important area for the NCI?
Certainly. I mean, we make use of some of these biomarkers, and the cancer field as a whole, which is largely supported by the NCI, has made use of MRD to influence clinical treatment for a long time. Probably started in acute leukemia, acute lymphoblastic leukemia first. But a lot of those things did start in blood cancers. That's the point I wanted to make, is that part of the reason you might be, well, why have we always done this for all cancers? Well, one of the things you need are options, and actual truly active options. And the blood cancer world has probably benefited from more active options for a much longer time than solid tumors have. It's only now that we have the opportunity to even consider in many solid tumors the fact that, oh my God, we might have not just one, but two things that actually have some effect on it. And only then can you build these more complex adaptive trials really that makes sense. I just want to make that point, that I think a lot of people agree that these are great ideas. it's only now that we can start to employ them.
Yes, and I agree. And that's because they're early endpoints being measured in the blood, which is really response to treatment in addition to the causal pathway. So you've made the point, I think, beautifully. So quick questions. We keep your questions short. We're going to keep our answers short so we can try and get to all of your questions. And then we'll have a final lightning round. Yes.
I think Maria Navas Moreno, CEO of Illumify Diagnostics. I appreciate the emphasis on the biomarkers being along the pathway of response. I think I find it a little bit ironic because we have so many therapeutics for which we don't understand the biology of it, and we have been using them for many, many years, and they keep getting approved. So really, more of a comment, but also a question. Then what are going to be the criteria and what are going to be the standards to really adopt these new biomarker, these early endpoints? We really need guidance for the new coming biomarkers, the new type of technology of endpoints and biomarkers. And I didn't get any clear answer or any in that direction from the panel. So thank you.
Yeah, thank you. So maybe, Rick, you want to start with that again. There's response, and then there is how you bin the patient. So the question is, what's the guidance going to be, or how can the biomarkers actually be used? What are you going to be looking for as criteria?
Well, I'm out of the agency, so somebody else is going to have
this gives you a chance to be freely speak.
Let me tell you my feeling on this. Obviously, it has to be where you're going to be using, and it goes back to the point that I'm talking about. Here again, if you're talking about an approval endpoint, that's going to have a much different level of evidence, okay, versus something that's being used to stratify patients in an exploratory fashion. So to try to say, what is going to be the total package here is I can't answer that in a day, basically, even. So that question is so expansive, it really can't be answered. At one point, could I just jump back to a former panel?
Yeah.
Okay, one of the things I'd like to talk about is guidances, and that came up on one of the previous panels. Remember, people were saying it takes 18 months to get a guidance through. Remember what we did in COVID, okay? Those guidances came out very rapidly. So push the agency to develop these guidances as rapidly as possible. They are not binding, okay? So they could be done as quickly as possible, and they should be living documents that are out there, that are being explored, and that are being changed as we go with time. Perfection is the enemy. The perfection is the enemy of good, so to speak. And we really have to push the agency in getting these guidance out as quickly as possible. I've been stressing this repeatedly. However, my voice has been silenced, and I'll leave it up to you guys to take up that mantra.
Question over here.
Yes, I'm going to talk about the basket of inflammatory biomarkers that's massively downregulated. Lots of diseases have this huge inflammation cascades that lead to cell death, and very, very well understood in terms of being in the disease pathway and causality. And yet, when you have this biomarker basket that gets downregulated massively, then you go into your pivotal trial and have to do some scale or something else that requires a very large, very long trial that may be prohibitive versus getting approval based on the biomarker data itself. Can you comment?
Who wants to take that one on?
I'll just make one quick comment. It's all about safety, efficacy, unmet need, et cetera. So each case has to be taken. But my view, until the biomarkers are absolutely gold standard and you're confident with a high degree of confidence that it's going to impact the clinically relevant measures, you need to keep measuring the clinically relevant. And the phase 3 trials are not prohibitively expensive. If you knew they had a very, very high probability of success, it's very cost effective. At some point, the biomarkers do become standard of care and become approvable, like hemoglobin A1C, cholesterol, et cetera. But you have to, I think, generate an enormous amount of data. PCR, MRD, these are things that have evolved to be confident enough to approve on.
And we all have to work on collecting the data collectively. Yes.
So on that point, all of the coming AI revolution stuff fails if we don't have really rich clinical data sets and if we lack linked non-clinical and clinical data sets.
That's right.
So, I've participated in running clinical trials before. We spend thousands of dollars on these patients, and yet we don't incentivize the collection of a $5 high resolution mass spec collect on blood or serum. So what can we collectively do to make that data possible, to collect that data and then make it shareable to the folks who are developing the tools that will make this possible?
Yeah, make it shareable for sure. And well, that'll be part of our lightning round, what we can all contribute.
Fantastic panel. I'm thinking back to Keli's talk initially and like the beautiful visualizations to sort of represent these are raw data that we're just interpreting. Both the trials that we're going to be running as well as the analyses we're doing are becoming more complex, and we understand them and believe we're at a future where we can use these right to benefit patients. How do we explain that better? Like if someone were to look at the x-axis on that outside of this room, is this a series of workshops Cersei puts on or some other thing to bring other people forward?
We're not letting Keli go back to the clinic and to finish his residency till he does that. And Rick is going to help facilitate making sure we have a workshop to gather all this data and show that it does work across the community of everybody, right?
Steve Goodman, Stanford. So I'm concerned about what I'll call the information risk when you make early decisions based on early biomarkers in clinical trials, or when it's approved based on preliminary interventions approved on preliminary evidence. What happens is the entire information gathering system is then dismantled. That is, when the clinical trial is ended, you can't get the information on the later effects, on the ones that don't have the same time scale. And the same thing happens at the regulatory level. Often you can no longer do a clinical trial or any sort of high quality study that would allow study. So how do you deal with this sort of regulatory information risk where our data gathering dissolves once these decisions are made, which can't be walked back? This happened in the COVID vaccine trials and also in the bone marrow transplant for breast cancer.
Yeah. Anyone want to,
I think that's an FDA question. And here again, I don't speak for the agency, blah, 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 blah. Okay, but the issue is, in all of the trials that we've given accelerated approval, when there's randomized studies, we do ask the sponsor and require them to submit their overall survival data, okay? We really want to take a look at that endpoint. Obviously, it's going to be very hard to collect safety data once patients go off trials.
What I'm concerned about.
When it's a single-arm trial, following somebody for for survival is questionable a benefit given how do you interpret that endpoint basically from a single arm trial. But we really have mandated, and many people have confused the issue that we were asking for an improvement in overall survival. We were asking to demonstrate that there wasn't a decrement in overall survival. And I think that's what your concern is. And I don't know what goes on in other therapeutic areas where the natural history may be so prolonged in some of these diseases that it doesn't make sense to, or is it practically impossible to do?
All right. So we're coming to the end of our time, and I do have our lightning round, but I wanted to say that I think the lesion in the system is clinical care. We have to require different kind of collection on outcomes in clinical care, and having that real world evidence would probably be transformative.
And it's how we analyze those, and maybe that's where AI can really help us out.
Yeah, so I'm sorry because we're standing between you and lunch, but I do have, this is a very quick lightning round. So this is the beginning of how we work better together. So as a community, to really think about the promise of early endpoints. And so I just want to say, although Rick, you can take it from any perspective you want, what is the one thing that you want to work on, to the one thing that you think is important from your organizational standpoint that you want to work on to advance this field? So Rick, I'll start with you.
Well, this probably has nothing to do with this panel, but I'll say it anyway. I really think that we have to take a look at our competitiveness in doing clinical trials in the United States. And I think we really have to leverage our existing technologies that we have, electronic healthcare systems, and work on embedding clinical trials in that system, have a portal in those systems that allow randomization within that, informed consent being collected. orders being written, and then that data being transferred into a cloud that then is available to all regulatory agencies. That is my dream of what clinical trials should look like in the next years. Okay.
I think that's what clinical care should look like too. Tony?
I think it will be hard to prosper with some of these more complicated biomarker ideas if industry isn't a little more on board. I think that perhaps I had the bad luck of working with a lot of unenlightened biopharma, but the fact is I have found significant resistance at good predictive, and I would say these are sort of, these early biomarkers are somewhat can be in line with predictive biomarkers. I would say that pharma doesn't see that benefit, and if it is seen perhaps at the CSO, CMO, CEO level, that has not been communicated effectively down through the entire organization, down to the level of people who are making decisions about clinical trials. So I'm afraid I'm a little more pessimistic about the state of biopharma right now.
Okay, but you're going to invest in better biomarkers and more studies to prove that, to make everyone more excited, right?
I mean, the NCI can do its part, but I'm just saying we're going to have trouble, and we do our part, but we're going to have trouble making this the general rule unless biopharma also sees the benefit.
Okay.
How many people here are from biopharma and are going to adopt this? Okay, thanks.
Could you answer that? Okay, Kate.
Yeah, so payers obviously have a lot of data. We often have the real world evidence that people are looking for. And almost every payer, certainly the big ones, all have a research arm that partners with life sciences companies. We do this at United and Optum. All the other big payers do as well. So I think that's the ways in which we can contribute, is to partner with academicians, life sciences, whoever, to generate some of the evidence that's needed.
And pay attention to PROs, right? Patient-reported outcomes. Keli.
Yeah.
I'll continue the theme. I think I want to continue thinking about the patient, thinking about data collection, and how can we incentivize data collection in this, big, beautiful system that we're thinking of in the future.
Hal?
Yeah. Maybe the way I'd say it is my history before medicine was in engineering, physics, and math. And when I think about the advances in physics over the last 150 years relative to what happened in medicine and biology, it's kind of comical. I mean, we have learned essentially how our universe works versus not really sure how you get diabetes. And I think we're about, with AI and massive at-scale data collection, going to enter a world where we have the equivalent in physics of theoretical physics. We're going to have theoretical biology. And that's why physics advanced so much, because there was a framework for the experimentalists to shrink their search space. And in the next, who knows if it's 10 or 20 years, we will be collecting data at a massive scale to understand the biology of the cell, the organ, and the organism. And when we have that theoretical biology, we're going to be making predictions like the physicists do that require technology so that the experimentalists can actually catch up. But we will be seeing things and transforming the industry. So my biggest hope is that we start collecting all this data at scale in a pre-competitive way and realize that we need to usher in an era of theoretical biology to transform the world.
Fantastic.
I'm going to have the last word, so you say it first.
Okay. Okay. Almost the last word. But anyway, so I would say that I'm a very short-term goal. The short-term goal is making sure that we collaborate as a community to show that these that continuous endpoints and can really be great predictive endpoints for the early cancer setting and would fit as robust early endpoints. We're going to work with Rick and the FDA to do a workshop by mid-year to show that this is possible. And the longer-term goal is to transform the clinic so that it is an engine of data collection and discovery to accomplish what Hal is talking about and everyone else has said. Yeah.
So as the patients, I do think I deserve the last word. I certainly endorse Laura's point. But what I ask is everybody here to adopt a little bit of the urgency that patients have. So we can talk about this, and talking about it helps, but let's everyone think about what we're going to do about it. And I did promise Hal that he could say one other thing.
I just wanted to say one thing, which is I've been doing this a long time and have had the opportunity to meet a number of incredibly inspiring people who've made a big difference. And I just want to say one of those people who's really changed the world in such a profound way in cancer is Rick Pazdur. And
I was liking it.
So can we put up the slide?
Thank you for everything you've done on behalf of all of us.
So we have. So here you go, Rick. Rick, you're on the big screen here.
Thank
we
you
want
so
to
much.
thank you for your service to the FDA
As you know,
as a global leader in drug development, and we hope that you will stay close. And
I will
continue to be
always
part of
be
it.
with you, okay? And you will always be in the silence
Doing it
of
all wrong, right? But
as people know, this was not something I had planned and is not something that I had wanted, but it is what it is. So let's just accept it. Thank you.
okay, thank you so much.